Hello everyone. Today I will lecture about subject and the main task of medical genetics, role of heredity in human pathology. So genetic is the science that deals with heredity and variation in organisms, including the genetic features and constitution of a single organism, species or group, and with the mechanisms by which they are affected. So as you can see in this slide, genetic variations cause inherited disease. Most of them is environmental disease like infection, traumas, and burns. And genetic disease is include cystic fibrosis, Down syndrome, sickle cell disease, and etc. disease. Genetic information include first one gene. This is basic unit of genetic information. Genes determine the inherited characters. Genome, this is the collection of genetic information. Chromosomes, this is storage units of genes, and DNA is a nucleic acid that contains the genetic instructions specifying the biological development of all cellular forms of life. So, human genome include 46 chromosomes, or we call this one 23 pairs. So, last two chromosomes is sexual chromosomes. In males, it XY, and in female is this XX. And the previous 22 pairs of chromosomes we called like autosomes. All genes in our body is responsible for own their function. For example, some of them is responsible for blonde hair, some of them for black hair, or genes is responsible for forms of eyes, forms of nose, and etc. things. Here you can see chromosomal structure. So each chromosomes we divide depend on size, big, small, and medium. Depend on location of centromere, we divide 4 types. If centromere located in the middle of chromosome, it would be metacentric chromosome. If centromere located near the one side, we call this chromosome submetacentric. If centromere located in the one side, we call this acrocentric chromosomes. And if centromere located in the upper side of chromosome or on the top, we call these chromosomes telocentric chromosomes. So, depend on size and depend on location of centromere, we divide these A, B, C, D, E, F, G groups. So, A group include first, second, and third pair, which is largest, depend on size, and first and third are metacentric, second is submetacentric chromosome. B group include fourth and fifth chromosomes, which is large, submetacentric, with 2 arms very different in size. C group include from 6 to 12 chromosomes. They are medium size, submetacentric. D group include 13, 14, 15 chromosome and medium size, acrocentric with satellites. E group includes 16, 17 chromosomes. They are small. 16 is metacentric, but 17 and 18 are submetacentric. F group include 19 and 20 pairs, which is small and metacentric. 21st, 22nd, and Y chromosomes are small, acrocentric, with satellites on 21st and 22 chromosomes, but not on the Y chromosome. So, human genetic disease and normal variations can be placed into one of 5 categories: monogene disorders, disease or traits where the phenotypes are largely determined by the action or lack of action of mutation. at individual loci. Multifactorial traits, disease or variations where the phenotypes are strongly influenced by the action of mutant alleles at several loci acting in concert. Chromosomal abnormalities, this is where the phenotypes are largely determined by physical changes in chromosomal structure like deletion, inversion, translocation, insertion, and etc. And chromosomal number, trisomy or monosomy, or chromosomal origin and parental disomy. And mitochondrial inheritance, disease where the phenotypes are affected by mutation of mitochondrial DNA. Also, congenital malformations is when congenital defects was of inner organs or parts of body. Here we show to you the mutation of chromosome. First one is deletion. During cell division, especially meiosis, a piece of chromosome breaks off, maybe an end piece or a middle piece. Inversion, this is a segment of the chromosome is turned 180 degrees. Translocation, movement of a chromosome or translocated to another segment of chromosome. Duplication is doubling of a chromosome. In this picture, you can see patient with Down syndrome in a karyotype. As you can see here in the 21st of chromosome, they are triple of chromosome. This is trisomy of 21 chromosome, and we call this disease Down syndrome. Now we will start about methods in medical genetics. First method is genealogical method. In genealogical method, we create the pedigree. So for this pedigree, we use special symbols. For example, this symbol we use for affected male. This symbol we use for female but unaffected. This symbol for male which is dead during the life, and this is mating between male and female. This is relative mating. This is pregnancy. So these symbols is for carriers. This is dizygotic twins. This is monozygotic twins. Also, we have in medical genetics inheritance. First way of inheritance is autosomal dominant inheritance. The locus is on an autosomal chromosome and only one mutant allele is required for expression of the phenotype. Affected males and females appear in each generation of the pedigree. Affected mothers and fathers transmit the phenotype to both sons and daughters. Example would be Marfan syndrome. In this picture, you can see autosomal dominant inheritance. Example is brachydactyly when finger is short than normal. Second inheritance is autosomal recessive inheritance. The locus is on an autosomal chromosome and both alleles must be mutant alleles to express the phenotype. The disease appears in male and female children of unaffected parents. Example would be cystic fibrosis. X-linked dominant inheritance. Affected males pass the disorder to all daughters, but to none of their sons. Affected heterozygous females married to unaffected males pass the condition to half their sons and daughters. In this picture, you can see this example. X-linked recessive inheritance. Many more males than females show the disorder. All the daughters of an affected male are carriers. None of the sons of an affected male show the disorder or are carriers. Example would be hemophilia. If the locus is on the X chromosome and both alleles must be mutant alleles to express the phenotype in females. In these two cases, you can see example and how many percent would be carrier, how many percent would be unaffected or affected. Last type of inheritance is mitochondrial inheritance. This type of inheritance applies to genes in a mitochondrial DNA. Mitochondrial disorder can appear in every generation of a family and can affect both males and females, but fathers do not pass mitochondrial traits to their children. Example would be Leber's hereditary optic neuropathy. Here you can see some congenital malformations. For example, here this is an undevelopment of ear. Congenital defect in the ear. Here you can see mongoloid eyes or anti-mongoloid eyes. Next method is cytogenetic method. This method gives us possibility to examine X and Y sex chromatin and chromosomes to establish karyotype. So with cytogenetic method, we identify chromosomal disease like Down syndrome, Patau syndrome, Edwards syndrome, Cray-Duchat syndrome, Turner or Klinefelter syndrome. In this case, for example, in Klinefelter syndrome, would be extra X chromosome in the last sexual chromosome, and it can be, for example, 47XXY or 48XXY. In the Cray-Duchat syndrome, karyotype would be 5p del. So its meaning in the fifth chromosome would be deletion. So then appear Cray-Duchat syndrome. In Patau syndrome would be trisomy of 13 chromosome. In Edwards syndrome would be trisomy of 18 chromosome. This was our first lecture. Thank you for attention.